Hey guys, so today I want to talk to you about an interview that I saw between Patrick Bet-David and David Icke, and the interview covered a whole wide range of topics, but what I specifically want to talk to you about is how David Icke is wrong about COVID-19. So here's a little disclaimer. This video refutes the claims about SARS coronavirus 2 made by David Icke during his interview with Patrick Bet-David. The intention of this video is to objectively refer the viewer to publicly available scientific data that clearly demonstrates SARS coronavirus 2 has been isolated and is a real virus. The views expressed in this video are not the opinions of the creator of the video. Please make your own conclusions after the video. So just so you understand what's going on, in case not all of you have a biological background, I'm going to explain what a virus is. So this is from a scientific journal. This is a diagram from 2009 of a coronavirus and how it infects a host. So this is the virus here. This is a viral particle, mostly proteins, and it surrounds a genome inside. And the genome is the genetic material, just like humans have genetic material, that controls sort of the life cycle of the virus. So really what happens is that these small particles will attach to a receptor in the host, okay? And once they attach to the receptor, in this case it's ACE2 for certain coronaviruses, once it enters, the viral case or the capsid dissociates and the genome exits and it takes over. So the virus hijacks the cellular machinery in order to produce new viral particles and spread the infection into other cells. So this is really how a virus works. It comes in, it depends on the host, and then it produces new particles with the cellular machinery from the host. So in this case, it would be a human lung cell. The virus would infect and then spread to other parts of the lung, and of course it infects other parts of the body as well, but I'm not going to get into that right now. So when it comes to coronaviruses, this is just how biologists classify different biological organisms. So coronaviruses are the order Nivovirales from the family Coronaviridae, and these are again just classifications. If you look at humans are of the order primates and the family Hominidae. So again, this is just the way you classify it. And here are some different types of coronaviruses that have been isolated. There are alpha and beta coronaviruses. So for example, in 2003, the SARS coronavirus responsible for that epidemic, that was a beta coronavirus. And then if you look at the Middle Eastern coronavirus from 2011, it was also a beta coronavirus. And the current one that's causing COVID-19, SARS coronavirus 2, is also a beta coronavirus. And this is from 2019 to 2020, right around New Year's when this was discovered. And so SARS coronavirus 2 stands for Severe Acute Respiratory Syndrome coronavirus 2, and COVID-19 is just coronavirus disease 2019. And so really the important part here to notice is that, again, I'm going to highlight the viral particle is separate from the viral genome, and it's really this particle encapsulating the genome. That's all that a virus is. So if we want to now identify this virus within somebody, one of the better ways to do it is to use this genome, which is completely unique to the virus. So the genes in this genome, and I'm going to get into more of this detail later, the genes in this genome are completely unique to the virus, and they can distinguish between the virus and the host. So what is a genome? So we've all heard of DNA, deoxyribonucleic acid, and this is what's inside of animals and these and plants as well. So these genomes, essentially, the way it works is you have these nucleotides which have nitrogenous bases, and those bases are complementary to each other. So the T's will stick to the A's and the C's will stick to the G's. So this is really two separate molecules, and they're stuck together at these A, T, C, G locations, and that's the way they always stick together. So in that way, it's a very replicative process. So when this DNA replicates, it always produces the same double-stranded molecule afterwards. Essentially, RNA is very similar. I'm not going to get into the complexities of what make them different. Again, you could see that there's a U instead of a T, so uracil instead of thiamine, but essentially what you get is you get this complementarity as well, and you really get these nucleotides that define a very specific, unique sequence to that specific RNA molecule. And RNA genomes are unique to viruses, so really they're only inside of viruses. Okay? So if we look at the coronavirus RNA genome, this is a linear representation of the genome here, and I'm just going to go into a few technical terms, but it's just important to describe the virus. It's a positive sense single-strand RNA virus. I'm not going to go into what that means. It's about 27 kilobase to 32 kilobases long, and so that's the length from this 5' to this 3' end, or from one end to the other. And this is all of the nucleotides, those little letters I showed you, A, C, G, U. So all of those nucleotides go across in a very unique order that are specific to the virus. Okay, so these are coronavirus genes, and the uniqueness of the nucleotide sequence is specific to the coronavirus, and it's not in humans, okay? And what this little depiction is here, I want to bring your attention to this RNA-dependent RNA polymerase, or RDRP, this red region here. And this is a gene that the virus encodes to produce a protein that helps the virus to replicate its own genome, essentially. So the reason this is important is because this gene is very specific to the virus. You will not find this in the human host. This only exists in the virus, okay? So just remember that for later when I talk about PCR. So the first claim David Icke makes is that there is no evidence that's been scientifically produced that COVID-19 exists. So I'm going to show you some videos here of him saying this, okay? So here's the first video here.
There is no evidence that's been scientifically produced that COVID-19 exists.
Okay, so that's his first statement there. You can see it's a very strong statement. He's certain that there's no evidence that's been scientifically produced. And now I'm going to show you another statement he makes. Okay, so here we go.
You don't believe that, though. You don't believe that coronavirus doesn't exist, though.
Of course. Well, I'll just say to you, produce the scientific evidence that it does. What I've been...
So here it's interesting. He again says produce the scientific evidence, and he very quickly changes the subject. So I don't know if that's just a coincidence, or if he doesn't believe himself there, or if he doesn't want someone to come up with the evidence, but I'm going to do it here. But I'm going to show you another couple things that he says first.
The problem is they never, and it has never, been isolated to show that that's what is causing this.
So again, he says it's never been isolated. I don't know where he is getting this, but I'm going to demonstrate to you that it has been isolated, and I'm going to show you exactly how this works, okay? And here's another statement he makes.
Now, in the end, what you're looking at is data. What I mean by that is not theories. You're looking at data.
Okay, so the reason I bring this up is because he claims to want to look at data, but really he doesn't look at much data, if any. He doesn't reference anything. If you notice, I've got references here. So anyway, he's not looking at data. It's a little bit contradictory. So I'm going to show you that there is scientific proof that COVID-19 exists. So here is a paper that was released February 20th, 2020 in the New England Journal of Medicine. And this was talking about a novel coronavirus that was isolated from 3 patients. And he, essentially, these 3 patients, they called this coronavirus 2019 nCoV for novel coronavirus. And it really was not just isolated using PCR. They did whole genome sequencing. So this is something that David Icke does not talk about. It's a high-tech method or procedure in order to find out the genetic sequence of these viruses. And 27, for anyone who knows anything about PCR, you cannot do it with something that is 27 to 32 kilobases. It's way too large. You need something like whole genome sequencing in order to really the best efficient way to find out what the genetic sequence of these viruses are. So these sequences were found out. They are in the databanks. And this is just an example of later on in that same paper where they actually give you, this is public information. You can go to these databases and find the genome sequences of the whole isolated coronaviruses from human patients, okay? They've been isolated. You can't sequence them without isolating them. They've been isolated and they've been sequenced, and they're even here showing comparisons. They're doing what's called homology comparisons between other viruses like this bat SARS coronavirus, okay? So you can actually see not just, as I'll show you a bit more later, not just the sequences themselves, but also a little bit more data comparing them to other genomes as well. So here is an example of those comparisons. This is called a phylogenetic tree, and here this is actually representative of 2,685 isolated samples of SARS coronavirus 2. This is what's causing COVID-19, and this is 2,685 isolated samples as of April 1st by this study in the UK. So really, this is this disputes what David Icke has said and shows that there are cases or isolated samples of SARS coronavirus 2. He wanted scientific evidence. Here's your scientific evidence. And I would like to see him try to refute this, because I really don't think he has the knowledge base to even do something like that. So he should not maybe be talking about this to the detail he is. So his first claim that there is no evidence that's been scientifically produced that COVID-19 exists, this is wrong, and I've just shown you that it was first isolated as 2019 nCoV and identified using whole genome sequencing, and as of April 1st, 2020, at least 2,685 complete SARS coronavirus 2 viral genomes have been isolated and sequenced around the world. Okay, so this is far away from zero like he's claiming, and really he's wrong in his claim here. So what concerns me about this is that he is making bold claims without reading the literature. He claims that he likes data and that he likes to do research, but he's not doing that here. So he is 100% convinced of these claims being true without any evidence. His evidence is very weak. I mean, we could talk about the video that Andrew Kaufman made, the psychiatrist, about PCR not being good to look at viral samples to distinguish them between human and viral genomes, or just to distinguish between human and viral genomes. That's a whole other topic. I could do a whole video debunking what that guy said, but again, he's not talking about the whole genome sequencing or other evidence. And as well, he contradicts himself, as I just mentioned, about the data because he's not looking at it. So he's speaking about biology without knowing anything about it. And I'm going to give you an analogy about basketball. Imagine you went to a basketball game and you were listening to a commentator who has never seen or heard of a basketball game before. So he would be describing events that he doesn't know properly, and really, if you are a fan of basketball, this is almost like painful for you to watch, because he wouldn't know what the hell he's talking about, okay? And in the same way, because I know biology, it's painful to listen to this man talk about things he has really no idea about. So in this way, he's kind of really spreading misinformation, and this is the very thing that he claims to be against. So it's a bit contradictory, hypocritical. It's not the type of thing I would really take as a good source of information on this topic, okay? I'm not talking about anything else he discusses. This is just about his views on coronavirus, okay? So David Icke claims that another claim that he makes is that RT-qPCR cannot distinguish between viral and human genetic material. I'm going to save you the clips because it's a bit long-winded as he describes how he feels about this. But essentially, this is the claim that he makes, and I'm going to just explain to you why he's wrong about this. It is a great way to distinguish between viral and human genetic material. So here's a test that was done. This is the detection of a novel coronavirus by RT-qPCR, okay? And really what they did is they used the PCR based on those whole genome sequences that have been released, and they can then design the procedure or this PCR to detect the virus. So I'm just going to give you a little bit of a background of what we're looking at here, just to orient you so you see. So this gene bank accession number, this is public information, okay? So I'm going to now bring you to the NCBI, or So the NCBI database, okay? And this is where they have biotechnology information. And essentially what you could see here is this is the severe acute respiratory syndrome coronavirus 2 isolate. So it has been isolated, okay? This is the isolated virus, and it shows you who has done it. There's been several people that have done it, and it shows you who's done it, okay? Now, if you look here at the gene bank, this is the number MN908947. This is the same number that I'm showing you here, okay? So just so you can orient yourself with what we're actually looking at. Now, if you go down and you scroll down on this page, you could actually see all the information about the whole virus. These are the amino acids that make up the proteins. So we're not going to get into that here because it's not relevant to the test. But then I want to show you this is the genetic sequence. So remember I said it's about 27 to 32,000 bases long, and these are those bases. This is the nucleotide sequence. So here you can see it goes from 1 all the way to about 29,900, okay? So this is the sequence. In order to get this sequence, it was isolated and then sequenced, all right? So now I'm going to just get into what this all means for testing for it. So here is RT-qPCR, or reverse transcriptase quantitative polymerase chain reaction, and this is used to test or to detect RNA from biological samples, okay? So reverse transcription is the first step. This is where you use these special enzymes essentially to turn the RNA into DNA. And really what this is doing, it maintains the sequence of the RNA, but now you're using a more stable molecule to do your analysis. So RNA is inherently unstable, and it's not a molecule you could actually expose to the types of environment, or at least the environmental conditions that are needed to do PCR. So by converting it to DNA, that's all you're doing, is being able to do this procedure. So really the way the procedure works is that you have a sequence of interest. This could be any sort of DNA, human, or the viral DNA, or any DNA here. And then what you do is you actually use a complementary probe that has fluorescent dyes on it. So this probe, these colors here are representing different nucleotides, and they only match up with the complementary nucleotide on the sequence of interest. So for example, this probe is not going to bind at another region on this sequence, nor is it going to bind to any other region on any other genome out there, because it's been designed in a way to only target this specific sequence of interest, okay? As well, the primer has been designed with the same specificity. All right, so this primer here, essentially it's just needed to start this chain reaction where this enzyme, this polymerase, will now travel along the sequence of interest, adding in nucleotides. And what this is going to do is this is going to displace the probe and release these dyes, and one of these dyes is actually inhibiting the other from producing color. So when you break them apart, now you can actually get the dye being produced. And in this way, you'll be able to see not only if you have the sequence of interest present by sending the probe in, but you'll also be able to see how much of it there is depending on how many of these reactions take place. So this is the quantitative polymerase chain reaction using the reverse transcriptase. And this is a great way that biologists can actually identify very unique sequences, specific sequences of interest, okay? So when you're using this to detect SARS coronavirus 2, again, this is your viral sequence here. This is the whole genetic sequence here with all of the genes. And I'll just remind you, it's about 2,900 and 900 RNA bases. And this is kind of what the sequence looks like, and it goes again to 29,900. Within this code, there is a code for this RDRP. And this RDRP, the RNA-dependent RNA polymerase, what I mentioned to you before, this is what actually has these very specific sequences for the primers and the probes that they use for the assays to detect for SARS coronavirus 2. So the key take-home here is that these are very specific sequences only present in SARS coronavirus 2. They are not in humans. So if you send these probes into human lung fluid, if you're getting a positive result, it means that the virus is present, okay? You can't really get a false positive. Let's just say you can't get a false positive from these tests in the human samples, okay? So this is the probe here, and I'm going to show you that I've actually copied this into this really cool tool. So you could see this is called a blast search, the NC National Center for Biotechnology Information blast search. And what this does is it takes the viral sequence and searches it through all of the biological data of all of the sequences out there and tells you if this aligns with any of those sequences in a complementary way. Okay, so again, this is CC, GT, CTG, and so on. So CC, GT, CG. So I've just copied and pasted this here, and now I'm going to run the blast search. So if this sequence actually matched up with anything in the human genome, you would see it in the blast search. You would see Homo sapiens or humans showing up in this search. So when we do this search, the results that we get should all be SARS coronavirus 2. So it'll show up in a second here. It's just searching the database right now. And I'll encourage you guys to play around with this. Go on YouTube, find videos that explain how this stuff works, because it's really fun. You could play around with it. You could find human genes, you could find other genes, and you can actually look and see where they match up with other genomes in the databases. So in this way, it's actually a lot of fun. It's actually taking a lot longer than it usually does. Normally this search is pretty quick. I might just try it again if it's not working for... Oh, here we go. So if you look here at this sequence, and all of the results are severe acute respiratory syndrome coronavirus 2 isolate. These are isolated coronaviruses, complete genomes, and you're getting 100% coverage. So really you're getting 100% sequence alignment, okay? And this is just for, if you look here, there is nothing other than SARS coronavirus 2. So this is really proof that this sequence, when you stick it into human or onto lung fluid, you're not going to get a false positive. This is going to only produce a result when you have the coronavirus, the SARS-2 coronavirus 2 present in the fluid. So again, if you have your sequence of interest as SARS coronavirus 2, you now put in these very specific probes and primers, and this is what is not mentioned by David Icke at all. I don't even think he understands this process at all when you hear him talk. But these are so specific that they will only adhere and anneal to and produce a positive result with the SARS coronavirus 2 viral genome. They will not produce a positive result with human DNA that is present anywhere in the lungs. That's not how this test works. So in this way, really, this is SARS coronavirus 2 specific, highly specific test, okay? And so David Icke's claim that RT-qPCR cannot distinguish between viral and human genetic material, again, this is wrong, and I've shown it very simply that these probes and primers are designed to target viral specific genomic sequences, as well that RT-qPCR can distinguish between viral and human genetic material. And it's actually a great assay for this, okay? So really my concerns are similar to the first one, but he's speaking 100% about claims that are not true, and he does not have evidence. And what really concerns me about this is that if you asked him, what is a reverse transcriptase? What is a Taq polymerase? Explain how PCR works. How are these dyes working? What is this assay, or what is this procedure? He could not do it. He is not the person qualified to talk about this. If you're going to get information about a molecular biology procedure, you need to speak to a molecular biologist. You can't get it from David Icke. I'm sorry. Now, he might have very good research or information about other topics, but this is kind of just low-hanging fruit for me because he's so wrong about this one. So he contradicts himself very often. And as well, previously in the interview with Patrick, he talks about the holographic nature of the universe. And the reason I bring this up is because he actually references science and says David has been talking about the holographic nature of the universe for so many years, and finally quantum physicists have proven that this is actually the nature of the universe and that he is correct, okay? And what bothers me about this is that he uses these claims, but he blatantly ignores the scientific evidence regarding SARS coronavirus 2 existing. And I really believe that it's because he thinks that it really pushes his agenda. So I don't want to get too much into his agenda as far as the leaders of the world and these secret societies. So I'm not going to talk about that here. But really, I think that he's allowing these biases and his agenda to cloud his reality. And if he's going to claim that science is proving that his holographic universe theory is correct, he has to believe that the science that I've demonstrated today and other science is true, or is proving that SARS coronavirus 2 does exist. You can't pick and choose what you want to believe. And so really he's kind of being hypocritical or contradicting the very thing he claims to be fighting back against, which is these, leaders pushing their agendas. Okay, so in this way, I don't know. I'm really questioning his credibility here, kind of like Patrick did throughout the interview. I'll also briefly bring up, just to defend this, the Koch's postulates that he talks about. This is a really good way or the best way to find out if you have a pathogen. You isolate it, you identify it, you infect it into a host, you re-isolate it, and you could reinfect it into a new host. And by doing this, you can really prove that this one agent is what's responsible for causing the disease in something. But the reason, and this is very simple, they explain it right here, okay? The reason that these scientists released this paper without fulfilling the postulates is because of time. They had time constraints. They wanted this paper to be released with the valuable information that it contained without going into all of these immunological and monkey experiments that are going to be done or are probably currently being done now that will prove Koch's postulates are true for SARS-CoV-2, but they mention it in their paper that it's of critical importance, but they just haven't gone ahead with it yet because of time constraints, and they wanted this information out there. So this is really a huge misinterpretation by David Icke on this point. So really my final thoughts are, please, when you hear people like David Icke or anyone, including myself, talk about something that they seem to know about, do your own research. Do not just trust it. We're not in a cult society. That's what happens in cults when one person talks and people blindly believe everything that they say. And this is exactly, again, the sheeple that he talks about. You don't want to blindly follow anyone, not David Icke, not Andrew Kaufman, not myself, not anybody. So please do your own research, okay? So that's really all I have to say. I hope you guys liked the video. If you did, I can debunk the claims by Dr. Andrew Kaufman, the psychiatrist, talking about how PCR leads to false positives and can't distinguish between human exosome DNA and viral SARS coronavirus 2 RNA. This is, again, low-hanging fruit. I have no problem debunking this. And again, I hope you liked the video, and give it a like if you liked it. Thanks a lot for listening. Take care. Bye.